Hello everybody, and welcome to this presentation entitled The Great Beauty of the Osteoclast. This story starts in 1974 when I began my internship with my teacher Alberta Zallone. This was the time when Steve Teitelbaum used to say, "Two ladies in South Italy, look what they did." It is a 40-year story of osteoclasts, and these are my very first osteoclasts in vivo. And these are my first osteoclasts in vitro. We were able to isolate and purify a large number of osteoclasts that allowed biochemical and molecular biology studies, as opposed to rat osteoclasts by Tim Chambers and Alan Boyd, which were too few and too contaminated with bone marrow cells to allow large-scale analysis, although they have been very important for single-cell studies. Osteoclasts are fascinating cells. They sit on the bone and resorb the bone matrix for two reasons: to perform bone modeling, to shape the bones during growth, and to perform bone remodeling, to replace old bone with new bone and keep our skeleton healthy. Osteoclasts are very easy to be recognized because they express a highly specific lysosomal enzyme called tartrate-resistant acid phosphatase, here in purple. The osteoclast is a multinucleated and polarized cell. Here you see the nuclei, the endoplasmic reticulum, many mitochondria, multiple Golgi stacks, and a high number of lysosomes that will be secreted by the cell. The osteoclast has also a peculiar plasma membrane with a basolateral membrane that faces the vascular stream and the apical membrane that faces the bone to be resorbed. In the apical membrane, you see here the peripheral area that is called sealing membrane and attaches the cell to the substrate, along with the nearby cytoplasm that is devoid of organelles and is called the clear zone. The sealing membrane forms the sealing zone. The inner membrane presents a number of irregular protrusions similar to microvilli that increase the cell surface, and it informs the so-called rougher border. The rougher border is the bone resorption apparatus of the osteoclast. The sealing membrane attaches the cell to the substrate, and if we look deeply, we see that it's formed by a number of short protrusions that we discovered in collaboration with Piercarlo Marchisio and called podosomes. Podosomes are like little feet, and probably they have some kind of lytic activity, because you see here that there is some disruption of the bone matrix, probably to expose the adhesion proteins and promote cell attachment. We discovered the podosomes in our very first osteoclasts that were observed to spread out on substrate and display a number of dot-like structures at the cell periphery. These structures contain a core of microfilaments, here stained with fluorescent ferroidin. At a higher magnification, you can clearly see these dot-like structures due to the orientation of the microfilaments perpendicular to the substrate. The molecular anatomy of the podosomes has been extensively investigated by us and others, and you see here that each podosome core is surrounded by rosette-like structures of adhesion proteins, here vinculin. And in this high magnification, you see clearly that the vinculin form a circle around the black core, here not stained with fluorescent phalloidin. The morphology of the podosomes is shown here, and you see that between the plasma membrane and the substrate there is some material which includes the integrin receptors, especially the alpha V beta 3. Podosomes are very dynamic addition structures that assemble and disassemble in minutes. They start forming as soon as the osteoclast attach to the substrate, increase in number and cluster throughout the cell, then they concentrate at the cell periphery, and during bone resorption increase in number and form continuous rings that we call actin rings, and that can be multiple during bone resorption. And indeed here you see multiple actin rings stained in green in a single osteoclast. Podosomes have many components. You see here actin microfilaments, actin binding proteins, adhesion proteins, integrins, and a number of intracellular molecules that, when the integrins are activated by addition to the substrate, signal the cells and change the cell activity. Podosomes are regulated structures. We demonstrated that they are pH sensitive. In fact, when we acidify the cells with sodium butyrate, the percentage of podosomes increase over control, whereas when we alkalinize the cell with bicarbonate, the percentage of podosomes decreased. And this has to do with the resorbing function, because acidification stimulates bone resorption and alkalinization inhibits bone resorption. Podosomes are also sensitive to the extracellular calcium concentration. In fact, when we increase the extracellular calcium concentration, podosomes disappear. And this makes sense because an increase of extracellular calcium concentration inhibits bone resorption. The rougher border is formed by a number of irregular protrusions. Here it has been demonstrated, especially by the group of Kalervo-Van Anen, that in the rougher border there are at least two subdomains. The external domain is called fusion zone because here intracellular vesicles, especially lysosomes, are secreted by the cell into the underneath extracellular environment that is called resorbing lacuna. The inner area is called uptake zone because here there is the internalization of the bone matrix degradation product that transcytose the cell and are secreted into the vascular stream through the functionally secretory domain that is opposite to the rougher border domain. The molecular mechanisms of bone resorption have been extensively investigated. Bone resorption starts with the carbonic anhydrase type 2 that accelerates the hydration of CO2 into carbonic acid. Carbonic acid is then dissociated and the protons are released into the resorbing lacuna through a vacuolar type proton pump. The bicarbonate are then exchanged through the basolateral membrane by a bicarbonate chloride exchanger. The chloride influxes the cell and then is released into the resorbing lacuna through a chloride proton antiporter associated with a beta subunit that is called OST1. Now here we have protons and chloride which acidify the resorbing lacuna, and this acidification solubilizes the hydroxyapatite. The solubilization of the hydroxyapatite uncovers the organic matrix of the bone that is made by 95% by type 1 collagen. Here now the lysosomal enzymes are secreted, and among these enzymes, especially the catepsin K, disrupt the collagen. And this release both the inorganic and the organic matrix product into the resorbing lacuna. All this not only to perform bone modeling and remodeling, but also to control calcium and phosphate metabolism, along with a number of other organs, including the thyroid and parathyroid glands, the kidney, the gut, and other bone cells, including the osteocytes. All these organs and cells contribute to the maintenance of normal circulating levels of calcium and phosphates, and these two ions are important for the activity of all cells of our body. Osteoclasts are very sensitive to inflammation, and inflammatory disease, their number increase and they destroy the bone, especially at the joints. This led to a new discipline that is called osteoimmunology. Osteoimmunology stands for the crosstalk between bone and immune system. And in fact, the osteoclasts are regulated by a number of pro-inflammatory and anti-inflammatory cytokines, shown here. And among the stimulatory cytokines, we focused our attention on interleukin-6. In fact, we had the chance to investigate mice overexpressing interleukin-6 into the circulation, and you see here that these transgenic mice are shorter than control and each bone segment is short. And here you see that in these transgenic mice there is an increase of osteoclast numbers, shown here in purple, along with the reduction of bone formation, shown here by the distance between these two levels that is normal in the wild type and almost disappeared in the transgenic mice. This induces bone loss as shown in this slide. And this bone loss is caused by a very complicated mechanism in which interleukin-6 not only inhibits osteoblast bone formation and stimulates osteoclast bone resorption, but it also induces a complicated regulatory loop in the osteoblast that leads to the secretion of the IGF binding protein 5 that we demonstrated here that increase osteoclastogenesis. Interleukin-6 is elevated in a number of diseases, including the Duchenne muscular dystrophy. In fact, we observed that both in patients and here shown in an animal model of DMD, that the circulating levels of interleukin-6 are increased. And in this animal model, you see here that there is an increase of osteoclast numbers, again here in purple, and bone loss. This led us to propose that in normal condition, there is a beneficial mechanical loading by the muscle on the bone and the balanced systemic cytokine pool that induce a normal bone remodeling with normal osteoblast activity and normal osteoclast activity. In Duchenne muscular dystrophy, we have reduced mechanical strength due to the muscle damage and a very high inflammatory status in the muscle that induce an increased systemic interleukin-6 and probably an imbalance of other systemic factors, and also the interleukin-6 stimulates the osteoblast to produce locally a number of pro-osteoclastogenic factors. So this induces a reduction of osteoblast activity and an increase of osteoclast activity, which lead to the bone loss. So probably counteracting the deleterious effect of interleukin-6, we could improve the bone and muscle phenotype in Duchenne muscular dystrophy. Osteoimmunology has been important to demonstrate that not only the osteoclasts are regulated by the immune system, but that probably they are themselves immune cells. In fact, through the osteoimmunology, the major mechanism of osteoclastogenesis has been discovered. This is the cytokine rank ligand that activates the receptor rank expressed by the osteoclast precursors. Rank ligand is produced by a number of cells, including the osteoblasts, lymphocytes, and other cells, and activates rank, which stimulates a number of signaling molecules that leads to the expression of osteoclast-specific genes. However, rank is not sufficient to fully activate this mechanism, but requires the cooperation of other receptors, for instance TREM and OSCAR, which are associated to typical co-immune receptors, DAP12 and Fc receptor gamma. Only the activation of these co-receptors induce a fully activation of the intracellular signaling to osteoclastogenesis. This led to the conclusion that the osteoclasts are themselves immune cells because express typical immune receptors. The question remains why we need an immune cell to resorb bone. Now, what I described to you so far deals with the bone resorption activity of the osteoclasts. But the question is bone resorption the only task of this cell? And my answer is no. In fact, there are data demonstrating that osteoclasts are involved in the regulation of hematopoiesis, bone formation, intraosseous angiogenesis, and the bone endocrine functions. These other functions of osteoclasts have been demonstrated especially through the investigation of an osteoclast disease that is called osteopetrosis. Osteopetrosis is also called marble bone disease because the bones are very dense. There is the obliteration of the internal cavity with hematopoietic and neural failures, and this bone is very fragile and is prone to fractures. There are two groups of osteopetrosis, the osteoclast rich and the osteoclast poor. In the osteoclast rich osteopetrosis, there is an increase of the number of non-functional osteoclasts due to mutations in genes involved in the mechanisms of bone resorption. In osteoclast poor osteopetrosis, there is no osteoclast formation due to mutations in pathways inducing osteoclastogenesis. Osteoclast-rich osteopetrosis has been very important for understanding the molecular mechanisms of bone resorption. In fact, in the human disease, we can observe mutations in the carbonic anhydrase type 2, in the A3 subunit of the proton pump, in the chloride proton antiporter OSM1 complex, and in the newly discovered gene PLEP1. All these genes are involved in the mechanism of acidifications, but there are mutations also in the cathepsin K gene involved in the degradation of the type 1 collagen that is responsible of a closely related disease called pycnodysostoses or Toulouse-Lautrec disease because the French painter was affected by pycnodysostoses. Here the acidification is normal, the hydroxyapatite is solubilized, but the organic matrix is not disrupted. The osteoclast poor osteopetrosis has been instead important for the discovery of the rank-ligand-rank axis along with the osteoprotegerine, that is a rank-ligand decoy receptors that prevent osteoclastogenesis. And osteopetrosis has been also important to discover the hematologic and circulating origin of osteoclast precursors, two parabiosis experiments in which the normal parabiont could cure the osteopetrotic model through the joint circulation. And this has been important for the cure of the disease because in this way it has been possible to demonstrate that bone marrow transplantation can rescue a normal bone resorption in affected individuals. And through osteopetrosis, we now know all the steps of osteoclastogenesis, starting from the hematopoietic stem cell, through the osteoclast precursor, that is a mononuclear cell, the immature osteoclast, in which several precursors fuse to form a multinucleated cell, which polarize and become an active osteoclast. And you see here some of the pathways involved in this process. Now, what about the regulation of hematopoiesis? Well, if you look at the bone marrow in the two forms of osteopetrosis, you will see that in the osteoclast-rich osteopetrosis, the bone marrow is fibrotic. whereas in the osteoclast poor osteopetrosis, the bone marrow is still hematopoietic, suggesting that the high number of osteoclasts, although not resorbing, can affect the microenvironment, stimulating the stromal cells to produce collagen. The knowledge on the mechanisms by which osteoclasts regulate hematopoiesis is still incomplete. What we know so far is that the osteoclasts secrete metalloproteinase 9, which releases the c-KIT ligand involved in the transition of hematopoietic stem cells from quiescent to proliferative niche. But at the same time, the osteoclast releases cathepsin K, which cleaves CXCL12 and stimulates hematopoietic stem cell mobilization. But osteoclasts also release calcium that through the calcium sensing receptor induce the hematopoietic stem cell engraftment at the endosteal niche. So you see here that there are different functions, and probably what is important is the balance between all these pathways to regulate a normal hematopoiesis. What about the regulation of bone formation? During bone remodeling, the activities of osteoclasts and osteoblasts must be balanced to keep an healthy bone. This is due to a tight crosstalk between these two cell types, with the osteoblasts that regulate osteoclast activity and the osteoclasts that regulate osteoblast activity. When the activities of these two cell types are unbalanced, for instance, the osteoclasts work more than the osteoblasts, then you can have a bone disease, for instance, osteoporosis, with a bone loss and a number of fractures. The mechanisms by which osteoblasts regulate osteoclast activity are known since many years, and to give you here just a very simple example, the rank ligand and osteoprotegerin cytokines are released by the osteoblasts. However, it is known now that also the osteoclasts can regulate osteoblast activity through at least 3 types of signaling molecules. Some molecules are released from the bone matrix during bone resorption, which requires an active osteoclast and regulate osteoblast activity. Other molecules require cell-cell contact and have bidirectional functions. Whereas, through the osteopetrosis, it has been demonstrated that also non-resorbing osteoclasts can influence osteoblast activity through the release of cytokines that Roland Baron called clastokines. And osteopetrosis, once again, was important to demonstrate the correlation between the osteoblast and the osteoclast parameters. In fact, you see here that when the osteoclast number is low, also the osteoblast number is low, and when the osteoclast number is high, also the osteoblast number is high. There are many factors involved in this crosstalk. For instance, from the extracellular bone matrix during bone resorption, TGF-beta, bone morphogenetic proteins, various growth factors such as FGF, IGF, and angiogenic factors such as VEGF and BFGF are able to regulate osteoblast activity. And you see here what is stimulatory and what is inhibitory. Bidirectional signals can be the semaphorin 3A released by the osteoblast that stimulates osteoblast and inhibits osteoclasts, whereas bidirectional cell-cell contact signals are related to the afferin F pathways. And here you see a number of clastokines released by the cells. Many of them are stimulatory, some of them are inhibitory. And we contributed to the understanding of the osteoblast anabolic role of the tartrate-resistant acid phosphatase, shown here when we cultured Calvaria osteoblasts with conditioning medium from cells that have been transfected with TRAP, you see here that the alkaline phosphatase activity, which is a marker of osteoblast function, is increased in the presence of TRAP. But osteoclasts also affect angiogenesis through the TGF-beta released from the bone matrix, which stimulates endothelial cells to produce VEGF. Endothelial cells are also directly stimulated by VEGF and BFGF released from the bone matrix, and by a number of angiogenic factors released by the cells as clastokines, including the VEGF itself, the bone morphogenetic protein 7, the EGF, and a number of members of the EGF family. Now, is this all? No. What else? Osteoclasts are involved in the hormonal function of a bone matrix protein called osteocalcin, which is involved in the control of energy metabolism. This has been demonstrated by the group of Gerard-Kersenti, who showed that the osteoblasts express receptors for insulin, which stimulate the production of a highly carboxylated form of osteocalcin secreted into the bone matrix. At the same time, the receptor inhibits the production of the anti-osteoclastogenic protein osteoprotegerine. In this condition, bone resorption is enhanced, the carboxylated osteocalcin is released from the bone matrix into the resorbing lacuna, and at the decided pH it is decarboxylated, enters the circulation and becomes a hormone. This hormone increases the sensitivity of fat cells to insulin and stimulates the beta cells of the pancreas to produce more insulin. What else? Well, osteocalcin is a hormone that has been demonstrated also to activate the Leydig cells through a specific receptor to produce testosterone. So, osteocalcin is important also for the control of male fertility. What else? Very recently, the group of Gerard-Kersenti has also demonstrated that osteocalcin is important for brain development and cognitive functions during embryogenesis and postnatally. During embryogenesis, the maternal osteocalcin crosses the placenta and prevents neuronal apoptosis and favors later in life spatial learning and memory. Postnatally, the osteocalcin produced by the bone crosses the blood-brain barrier and induces an increase of adult neurogenesis, an increase of serotonin and dopamine, and a decrease of GABA neurotransmitters. All this prevents anxiety and depression and favors spatial learning and memory. In conclusion, I showed that the osteoclasts are bone-resorbing cells that contribute to bone modeling and remodeling, which is important for the maintenance of a healthy skeleton and for the regulation of calcium and phosphate homeostasis. But they also have numerous functions beyond bone resorption, especially through their clastokine kit. In fact, they regulate hematopoiesis, bone formation, intraosseous angiogenesis, and the hormonal functions of osteocalcin. Leading to the conclusion that osteoclasts are no longer stupid cells that just destroy the bone. This definition is by Steve Teitelbaum in 1985, but they became smart cells with a number of other roles, and this is my definition in 2014, underscoring the great beauty of being an osteoclast. I want just to acknowledge the people of the skeletal disease laboratory headed by Nadia Rucci and of the bone biopathology laboratory headed by myself, who contributed to the understanding of the osteoclast function, and all our financial support. Thank you very much for your attention.